So this is a really interesting field to follow, and it's had its share of Nobel Prize-winning discoveries over the last 10 years. If any of you haven't heard of CRISPR-Cas9 yet, we're going to talk about it. And before we talk about it, I think it is, especially for educational purposes, important to go over what was there before it, which helps provide context not only for why it's so powerful and interesting, but also how these fields of research are advancing. And we're not just going to do that for these genome editing techniques. We're also going to do that for more recombinase-based strategies, which we might not get to this time. So I won't spend a super amount of detail on the history, but it's useful to know that when thinking about editing the genome, a lot of people's orientation were around enzymes that cut DNA at a particular region. And that's what a nuclease is. So you could have a meganuclease, something that's really large, so that could be an unwieldy protein to express in the first place. And it could look for a very long, but probably somewhat specific or somewhat limiting kind of sequence, and then make a cut. And maybe it wouldn't just make that cut anywhere. And these are things that, you can think about, well, if you want a particular kind of modification, if you wanted to hypothetically mess with an RBS, even though most of these are oriented towards eukaryotes, then that might not be the right tool for you. So then the next iteration were these zinc finger nucleases, because zinc fingers are domains that tend to bind groups of 3 bases. And so almost like a codon, but it doesn't have to be a codon, because the word codon sort of implies that it's encoding a protein sequence and on RNA. So you've got these triplet nucleotides, and so you could actually fuse different zinc finger domains, thus getting something that's more specific. Because frankly, if you're just targeting the sequence, say ACG, in a genome like a human genome, it's over 1 billion bases, you're going to see ACG a lot. And so the longer you make that recognition, the more precise you're going to make your cut. And so there's this FOC1 nuclease that people fuse to these, but now you've got this like multi-domain fusion protein. And it's okay, but you're kind of stuck with these groups of 3, which mean that if you actually want to target something more specific than whatever, by the way, you don't have every possible set of 3, most likely, although some people might have. There was a lot of work in the early and mid 2000s to try to elucidate the protein DNA interactions so that you could reprogram what any of those were. And the same thing was true for talons. These actually came from plant pathogens, transcription activator-like effector. And so there were tail proteins in the pathogens, and the fusion to the FOC1 nucleus makes it a talon. And they were superior because these domains were not only, I think, smaller, but there was an actual code in terms of specific recognition of a domain to one nucleotide, which at the individual level meant then that you could combine them to get exactly targeting to the sequence that you want. And for a while, actually, after CRISPR-Cas9 came out, and probably still in some instances, you might prefer a talon over CRISPR-Cas9. So this technology, some of these technologies are not outdated and necessarily entirely superseded by the one to the right. But something very different about CRISPR-Cas9, which very interestingly emerged as a natural sort of innate immunity, or I should say it's an adaptive immunity strategy present in some bacteria to recognize virus sequences, to remember them, and then to next time the virus comes back to cut that sequence that came from a virus. So people started to notice this, in the mid to late 2000s. There were some papers that actually looked at yogurt bacteria, and I think Danisco and DuPont actually had some early interest and recognition that there were these repeating sequences. And the repeating sequences were the spacers in between each different memory element or sequence of DNA that came from a virus that was remembered. And so that's just a little bit of history, and I've certainly skipped the, or maybe I'll mention next the major players and the discovery of CRISPR-Cas9. But before I get off the rails here, something just to note about this and what's drawn here is that CRISPR-Cas9 is, the Cas9 protein is directed by guide RNA. So it's actually an RNA sequence that tells it where to make its cut. It is naturally a nuclease. It's not some fusion artificially created. And that's really powerful because now you're just giving it an RNA sequence. You don't have to work, and it's just one protein. This Cas9, you give it one RNA sequence, it goes there, makes a cut. You give it 10 different RNA sequences, it goes to all 10 of those places, makes cuts. And maybe most interestingly, Cas9 started to work in just about every organism that it was tested in. And that universality was more than a game changer. Honestly, a game changer is a huge understatement here. So you get with these cuts, in regardless of which case you use, a double-stranded break, and you can have the break effectively be repaired through non-homologous end joining, or an NHEJ mechanism, which also results in some indels usually at the intersection. So you don't get a clean, if you got a cut and you had two DNA sequences like this, you don't just get the sequence that you'd expect for them stitched back together. You get some kind of variations in the middle as the cell is trying to figure out, kind of calling an audible, trying to figure out what bases go there. And there's homology directed repair, which often relies on you providing some kind of a template, which is maybe what you want to do in a lot of cases. And you give the template, and this is a common theme that you'll see throughout this genome engineering section, some what we call flanking regions of homology. Those are sequences that match exactly the native genome sequence on either side. And the cell will look at these homologous sequences and want to stitch them together. So what I'm going to do next is just briefly cover CRISPR-Cas9 and then come to a stopping point. It means I might run a little bit over. But just on the topic of Cas9, I think I should go over a few more things. So I just mentioned that targeting is driven by the sequence of a guide RNA. And so in yellow here is the illustration of the guide RNA. And this is the target sequence that you want to cut. This is illustrating a Cas9 protein in blue faded in the background. And you've got your genomic DNA, and here you've got some kind of unwinding of the duplex here. And there are these red sequences which illustrate the fact that Cas9, at least its original version, and still most of its derivatives, had its own slight hiccup or constraint. It needed a PAM sequence, which is a protospacer adjacent motif, usually 3 nucleotides, and usually NGG, at least originally. And so N obviously being any base, but then 2 Gs that had to be next to each other and within a certain distance of the sequence you wanted to cut. So at first, you could make cuts just about anywhere and really tailor to where it is, but you still have to have this 2 Gs and something else nearby. So that was a slight constraint, but not too big of a deal. Although, you know, if you imagine that what you want to actually do is, say, revert the diseased allele for cystic fibrosis or sickle cell anemia to exactly what the wild type, the healthy variant is, you might not find your 2 Gs right there. That could be a showstopper. So that's just one reason why we care so much about constraints like this. Cas9 is also kind of a big protein. There are smaller versions that have come out since, but they're also not the smallest. Let's see. So you've got your Cas9. It doesn't do anything until you introduce a guide. Your guide RNA has some other features, including the target sequence. You get this complex where now the guide actually goes first to the Cas9, not to your DNA, and then it helps find. It brings the Cas9 to where it needs to go, makes a cut. And yes, this is illustrating that you can then provide a gene. And so we'll talk about next time what your donor gene might be, whether you want to do an insertion or maybe whether you just want to select for a deletion, depending on what your objectives are.